This is Component 3 in Class Question Book, Question 5. Right, Type 2 diabetes. And we've got a seemingly quite complicated diagram. So, where are we on this diagram? We are in the kidney. And we are, if I draw a Bowman's capsule, and you've got the proximal convoluted tubule, loop of Henle, distal convoluted tubule, then collecting duct. So we have blood capillaries, so we've got the glomerulus, afferent, arterioles, and then we've got blood supply in contact with blood capillaries in contact with all of this, the vasa recta. And so this is our blood capillaries, and the lumen. The lumen there is inside this proximal convoluted tubule, and these are the cells lining this proximal convoluted tubule. So that's where we are. And these epithelial cells, we've got some proteins in this cell membrane. We've got a co-transport protein, which is there. And we've got a GLUT2 protein, that's there. And we've got a pump as well, and the other one is a pump. So this relies on your knowledge of co-transport, and part 2 of this is the co-transport question, so we'll actually do that when we get to part 2. But for part 1, describe a biochemical test that could demonstrate the presence of glucose in urine, and why the test can only provide semi-quantitative results. Well, just reminder of your test for reducing sugars. So this is our test for reducing sugars, which is add Benedict's reagent and heat. So Benedict's reagent and heat, that's going to be the first mark. Then we've got, there's a colour change. And that goes to brick red. And so we've got 2 marks there. We've got the name of the test with what you do, and then the colour change. And then the last bit of the question, why this test can only provide semi-quantitative results. That means that you can get an indication of how much sugar there is, how much glucose there is, but it's not really a proper, it doesn't give you a proper number, like on a scale. So we can say that the colour change, gives an approximate, value for glucose. So for example, it kind of goes, green, then it goes to red. So there is a range of colours, and those are in your notes. But this is subjective, which means it's just the user's indication of the colour change rather than it actually being on a proper scale. Explain how glucose is reabsorbed from the filtrate into the blood, even at very high blood glucose. So this is saying our glucose is here. This is our glucose. How does our glucose get into this blood over here? Even if there's a high concentration of glucose in the blood. So even if it's against a concentration gradient, and we're going to have to talk about these three proteins we've got in this epithelial cell. So the first thing that happens is we have got the story starting actually on the other side of the cell from where the glucose starts. So we've got this carrier protein, this pump, and we've got sodium being pumped from the epithelial cell into the capillary. So we've got sodium ions actively pumped out of epithelial cell. I'll mention what it's like by the carrier protein into the capillary. So that's the first thing that happens. Then if we're pumping sodium out, we are reducing the sodium inside the cell. So reducing that sodium concentration inside the cell. So this reduces the sodium ion concentration in the cell. So now we've got a concentration gradient for sodium. So sodium is in the filtrate here. So this is our filtrate. This is our blood, and this is our epithelial cell. We've now got a concentration gradient for sodium. So sodium wants to, or will have a tendency to move into the cell. But we can only get in via this protein that we'll explain in just a second. So we need to just say about this concentration gradient. So sodium ion concentration now higher in the filtrate than the cell. So that, there's a gradient. So then sodium is co-transported. So sodium can get in through this SGLT2 protein, but only with glucose. So sodium can get in and glucose can get in, but only at the same time. So co-transported with glucose into the epithelial cell. So sodium can only get in if it's got a glucose as well, and that's really important. So sodium can only follow its concentration gradient if there's a glucose attached, and that's how glucose effectively gets transported actively into the cell. And so now the glucose concentration in the cell now higher, and it's actually higher than the blood, so higher than the blood capillary. The plasma in there, that's right, plasma, just to make sure it's dissolved in the plasma. And so glucose will now move out of the cell down its concentration gradient into the blood, but it's going to have to go through the one that I've marked in green here, the GLUT2 AO2 carriers just by facilitated diffusion. So glucose moves out of the epithelial cell by facilitated diffusion. And let's say the name of the protein via the GLUT2 carrier protein. And how does this process maintain itself? Well, the blood is moving, so this arrow here indicates the blood is moving, and that maintains that concentration gradient for glucose to eventually move out. So circulation of blood maintains plasma concentration of glucose lower than the cell. So it's a great question really, this, because you've got your knowledge of co-transport, which you should have memorized, and you're relating these proteins to your existing knowledge. So when it says, you know, things are diffusing or co-transport, you can associate these with your knowledge. So there's nothing new really needed, you just need to apply it to that diagram, which is classic AO2 territory. Over the page, a lot of writing again, and it's telling us about hyperglycemia. So hyper is high, hypo, if you see it anywhere, is low. So too high glucose levels, and it could cause damage to your eyes. And we are trialling the use of an inhibitor of the SGLT2. So an inhibitor of that. So on our diagram here, if I put a big I there for an inhibitor, which will stop that from working, and that can control blood glucose levels. So using the information provided above, so we need to somehow use that information above and on page 12, which is our I know it's page 16, this is page 12 of the original exam paper. Explain how the inhibitor could be used to treat type 2 diabetes. This is actually easier than what you think. Tell us what the inhibitor does and what would happen to the glucose. So the inhibitor would reduce the level of glucose co-transported, or you could say absorbed, or reabsorbed. So sodium wouldn't be able to follow its concentration gradient through this protein because it's been inhibited, so glucose wouldn't attach to it. And why does this help? Well, so glucose now would be lost in the urine. So glucose would be lost in the urine. Let's put urine on. So glucose would stay here and out. So glucose would go down the collecting duct and to the bladder. Suggest how the use of the inhibitor could also be used to help obese people. So this is AO2 again. So we're just using this information. If we've got glucose going out into the bladder, then it's not available for respiration, storage, use. The body would have to find its glucose from elsewhere, so it wouldn't be as efficient using glucose. So less glucose available for respiration. Now, other substrates, e.g. body fat, would be used instead. So that in itself is enough, but we could say less excess glucose. So if we've got lots of glucose in here, less will be reabsorbed and stored as fat. And so three things you can really say for those 2 marks. So a great question, 11 marks, showing that if you've done your revision and you know your stuff, all you need to do is just apply it to a new situation, and the more practice you do, the better you get.